Welcome friends. Now we are going to start a new topic, that is metabolism of glycogen, where we would discuss about the synthesis, breakdown and regulation of glycogen. In this video we are only going to discuss about the breakdown of this glycogen, okay? And about the introduction of glycogen, the polysaccharide, you can go to my YouTube channel and see the introductory video on glycogen. I would put a link in the description box. So let's start. What is glycogenolysis? First we split the term. This is glycogen polysaccharide and lysis means breakdown of this glycogen. How it happens? First we see some definitions. A process by which the primary carbohydrate stored, so this is a storage kind of polysaccharide, in liver and muscle cells is broken down into glucose. For what? To provide immediate energy and to maintain blood glucose level during fasting. So two motives are here: to provide immediate energy and to maintain blood glucose level. Second, we can say a process comes into play during period of lack of glucose. So this is shortage of glucose, and we can say hypoglycemic episode or accelerated glucose utilization. That is, we can say example, oxygen deprivation in case of anaerobic glycolysis. Or simply we put breakdown of glycogen, that is containing n number of glucose residues, to glucose 1-phosphate plus glycogen. This glucose 1-phosphate is the glucose residues after removal of the one residue from glycogen molecule, which is now containing one glucose residue less than n. So this is n minus 1. And next here is initiation of glycogen breakdown. So let's move to the process, how it happens. This is initiated by glycogen phosphorylase. So this is enzyme. It catalyzes the rate limiting step in glycogenolysis. Acts on the alpha-1,4 linkage, glycosidic linkage. So this is the important point. It is acting on alpha-1,4 glycosidic linkage at non-reducing ends through attack by inorganic phosphate and removing terminal glucose residues as glucose 1-phosphate. This is called as phosphorylasis, not hydrolysis. There is a difference between these two processes, and you would see. Difference is that some of the energy of the glycosidic bond is preserved in the formation of phosphate ester in case of phosphorylasis, and in hydrolysis there is not such kind of energy conservation. Cofactor here is pyridoxal phosphate for this enzyme glycogen phosphorylase, and there is a picture for this, the diagram showing the hydrolysis and phosphorylasis same time. You can see that. Now removal of glucose, there would be a picture where you would see that what we have studied is going actually. So this is that. You would see it yourself. So let's move to the next slide. Now the second enzyme is debranching enzyme. So this enzyme is coming to play now. The phosphorylase comes to a standstill. After some time, the first enzyme, which is the phosphorylase enzyme, would stand for some time, where 4 residues from an alpha-1,6 branch point, where there it finds a branch point 1,6, it would stop 4 residues away from that. You can see here. This is our, we can say, this is our branch point here. When this enzyme phosphorylase thinks that now I have reached 4 residues near to this branch point, I must stop. So it stops. This is followed by another enzyme, debranching enzyme. Okay, here debranching enzyme takes over, which is also called as alpha 1,6 to alpha 1,4 glucan transferase. Why this is called as this? We can say this is transferring alpha 1,6 glucose residue to the nearby branch which is containing alpha 1,4 linkage. This enzyme is containing two activities. First is transferase activity. The enzyme is going to shift a block of 3 glucose residues from the branch point to a nearby non-reducing end. Let's see that. This is our branch point. Phosphorylase enzyme has broken these glucose residues. Now three residues are there. So this transferase would transfer these 3 residues to the nearby branch point and would join it. Okay, now there is one residue remaining which is in linkage with 1,6. We would come to that now. So second is glucosidase activity. This would remove the single glucose residue which is remaining at the branch point. So this is now remaining at the branch point. This is going to remove this, and this has been removed. What we are ending with? This thing. This is our new glycogen chain which is containing no any branch point, and this is a perfect substrate for our initial enzyme glycogen phosphorylase, so this can act upon it now. Now we have produced this glucose 1-phosphate after glycogen phosphorylase reaction. What to do with it? This is the end product of glycogen phosphorylase reaction. Okay, it is converted to glucose 6-phosphate by phosphoglucomutase. So this enzyme make a shift of phosphoryl group from first position to 6th position. Okay, we know glycogen is present in muscles as well as liver, so we talk about them. In muscle, glucose 6-phosphate formed in muscle can enter glycolysis. For what purpose? To produce energy for muscle contraction. That is okay. In case of liver, there is a bit difference, you can say. Glycogen breakdown has a different purpose here, and that is to release glucose in the blood during low blood glucose. But why? Because this liver is responsible for homeostasis. Okay, but how we are doing this, and why muscle is not doing this? That why muscle is not delivering glucose into the blood, and why liver is delivering glucose into the blood? Because of the special enzyme which is possessed by only liver and not the muscles. It is also present in kidney, but for the context of this video we are only talking about this liver. So this is glucose 6-phosphatase. Remember, gluconeogenesis, third bypass reaction, where glucose 6-phosphate is converted into glucose by the action of this very enzyme. So here comes the question: why muscle and adipose tissue do not show gluconeogenesis? So very simple answer is that lack of this particular enzyme, that is glucose 6-phosphatase, and thus do not contribute glucose to blood. So I hope it would work. Thank you.